It's my great pleasure to introduce Dimitri Vandeville, who's our speaker for today. Dimitri is a professor of bioengineering at the EPFL, and he is an engineer and has expertise in computational neuroimaging. He's focused on cognitive and clinical neuroscience. He uses various tools. He's known for his work in wavelets as well as sparsity and graph signal processing and dynamics, among other things. So we're looking forward to hearing his talk today on graph signal processing for computational neuroimaging. And with that, Dimitri, I will hand it over to you. Okay.
Thanks a lot, Vince. Thanks a lot for the introduction and organizing this together with Armin. I really appreciate.
Sure.
In these difficult times to bring the community together. So today, as you announced, I will talk about graph signal processing, which is an emerging field in signal processing, and that I think is particularly interesting to be applied to computational neuroimaging. So the structure of the talk will be as follows. I will start with a quick introduction on network neuroscience and what it brought us the last 10-15 years in terms of how we can look at the systems level organization of the human brain. And in particular, I give a few quick highlights how more recently people are trying to combine structure and function and analyze them together instead of separately. And then I will go to a little let's say, more theoretical introduction, but I hope quite intuitive and quite fast, about graph signal processing and the basic ingredients that we need to build up our new operations that I want to apply later to the neuroimaging data. And so we'll introduce concepts like the graph shift operator, what's the importance of the spectral domain, and how this can be used to do filtering of signals on the graph. And we'll see that the central role here will be played by the graph Laplacian, and I try to explain why. And after that, we'll move on to the main part, which is revisiting resting state fMRI, but now using graph signal processing to have new ways of processing our spatial activation patterns. And we will, among other things, quantify how much activity is aligned with underlying structure, and GSP provides for that great tools. In a second main part, there will be also a further generalization of GSP to what I will call high-resolution brain graphs, where we will get from a few hundred nodes to basically almost 1 million of nodes, and then generalize our GSP operations on that. And to conclude, some more ongoing work, but that's of course always where the excitement lies, which is about making GSP community aware. Okay, so let's quickly set the stage. So network neuroscience has been tremendously successful the last 15 years. And if I kind of summarize it almost a bit like a cartoon, we always start off typically with our favorite brain atlas. That's defining the nodes of our graph, and then we need to come up with a way to get the edge weights. So typically either they are obtained from structural connectivity, so by applying tractography to diffusion-weighted MRI, and that leads to edge weights that characterize the strength of neural connections, which, if you put them all together in a matrix, constitute the so-called structural connectome, or the wiring diagram, as it is also usually called. But one can, of course, also look at functional information, and then by using correlation to estimate statistical dependencies between time courses, we can get edge weights as a measure of information sharing, which constitute the functional connectome or the communication diagram. And you can consider your structural or functional connectome as an adjacency matrix of the graph and apply many fancy graph analysis. And that has been done to get insights into the organizational principles of the brain. Now, more recently, the community has looked into many ways to integrate structure and function. And I'm just going to give a few highlights because this is a very active field of research the last couple of years. Of course, the very first thing you can do if you have a structural and a functional connectome is to look at the correlation between the edge weights or between derived graph features. That, of course, has been done, but it's rather descriptive. And since then, many other approaches have been tried. There is, for instance, the fusion of analyzing jointly structural and functional connectivity. There is tract-weighted functional and dynamic functional connectivity. Or in a different category of techniques, we have seen a whole repertoire of neural models that are defined on a structural connectome and that can then be used to simulate activity, which can be compared against the empirical one. We've seen a whole field being emerged around brain gradients and connectopies, also brain activity eigenmodes as sometimes called, which are using functional connectivity from which gradient maps or principal components have been derived. Also more recently is the use of structural connectivity to then look into principles of control theory and network communication to try to explain how function could arise and be explained on top of these structural connectomes. And then the last highlight is on the so-called connectome harmonics, which are in essence an eigendecomposition of the graph Laplacian from the structural connectome. And this last work bears most similarity with what I will show you today, and I will make a further connection later on. But before that, let me give you the basics of graph signal processing and what we will need for our later application of it. So just to set the stage. So we will be talking about undirected weighted graphs with n nodes, and thus they are defined by a symmetric weighted adjacency matrix A with weights defined in the notation you see here. And that from that we can also derive our diagonal degree matrix D and the total edge weight that we will define as a scalar m. So that's the classical graph definition. On top of that, we will also consider a graph signal, which is actually a vector of length n that attributes a value to each node. So we have our graphs and our graph signals. And then we need to come up with our choice of a graph shift operator that I will call S, and it will be a matrix as well. So graph shift operator is a linear operator that will is defined on the space of graph signals, such that each entry of the shifted graph signal is a linear combination of the input entries, and typically only involving the neighbors. So the adjacency matrix itself can be considered as a graph shift operator. But more commonly, people consider the graph Laplacian, which is defined as the degree matrix minus the adjacency matrix, as their choice of the graph shift operator, or in many cases also the normalized version of the Laplacian, which is the symmetrically normalized choice that we will make. And very importantly, we will also define the eigendecomposition of the graph shift operator S in the usual way, and we denote the eigenvalues by lambda and the eigenvectors by the vectors u, as you can see here. Now, the choice of the graph shift operator is very important because in GSP, this is inducing our graph Fourier transform. We're basically using the eigendecomposition of S to define a transform that brings a graph signal X into the spectral domain. And since this is a unitary transform that is defined by U, we can get back with a perfect reconstruction to the original signal. That also means that actually if you apply the graph shift operator to a graph signal, you could look at that operation in the spectral domain by a weighting of the spectral coefficients of your graph signal by the eigenvalues of the graph shift operator. And once we have our graph shift operator, we can now introduce more general graph operators H that we want to be shift invariant under our graph shift operator S. So it means that H and S need to commute. And the choice that you can then make is actually the family of matrix polynomials of S. And due to the Cayley-Hamilton theorem, the degree of that polynomial can be limited to n-1. In essence, it means that any graph operator H that is shift invariant under S can always be written in a spectral representation where in the spectral domain defined by the graph shift operator, it reverts to a simple weighting of the spectral coefficients of your graph signal. And that's exactly as you can see it here when we apply the graph operator to graph signal X, we can, by this identity here, we can interpret this as the graph Fourier transform applied to X, and then we just do a simple weighting by the spectral weights that characterize the graph operator. Let me try to represent this a little bit more intuitively, maybe. If we start off with an original, in the graph signal in the original vertex domain, we will apply our graph Fourier transform, such we get our graph spectral coefficients. And then we can apply our filtering operation by simple weighting of the spectral coefficients in the spectral domain. So the matrix H hat here is a diagonal matrix. And we get back to get our filtered graph signal in the original domain by the inverse graph Fourier transform. So of course this reminds you probably to the celebrated convolution theorem, and we're basically applying our filtering in the spectral domain. But if you think about it a little bit closer, we actually crossed a very big difficulty because the connection between the graph signal and the filtered graph signal in the original domain is non-trivial. And that is because by our choice of graph operators that are shift invariant with respect to X, we actually have applied through the easy spectral implementation a very complicated shift variant filtering in the original domain. And you can see this by constituting the matrix H you see here at the bottom, which directly links the filtered signal against the original one. And that matrix H is nothing else than U times H hat times U transpose, which on its rows contains the impulse responses that are not necessarily shift invariant, but that will adapt to the non-trivial structure of the graph. So why then is the graph Laplacian the choice we commonly make in GSP? Well, you probably know that the graph Laplacian is a kind of second order derivative defined on the graph. If you actually think about how you would do on a Cartesian grid a second order derivative with finite differences, you would indeed subtract the central sample from its two neighbors. And that classical case can be visualized by considering a cycle graph, for instance, which is actually a graph representation of a one-dimensional signal with periodic boundary conditions. So let's say we have here eight samples which are connected in a cycle graph. So this would be the adjacency matrix, and here we have the Laplacian matrix of this simple graph, which of course connects with +1 the node with itself and -1 with its neighbors. And if you compute the eigendecomposition of this Laplacian, what you get is actually exactly equivalent to the discrete Fourier transform. You get pairs of orthogonal basis vectors with increasing frequency. So here you see the eigenvalues, and here you see the eigenvectors, and you see indeed that you get like a DC component, and then you get two slow oscillating basis vectors, which is the first fundamental frequency, etc., etc. And that's why, in general, when we will define the graph Fourier transform through the Laplacian, we will consider the eigenvalue, lambda, as the frequency and the eigenvector as the graph Fourier vector. So a few prototypical spectral windows that are often used in GSP. So let's say we have a graph and this is our Laplacian spectrum as you would get it. Now you can come up with an ideal low-pass filter, for instance, or an ideal high-pass filter. Or you could take inspiration from diffusion processes and use a diffusion filter. Or you can implement actually the Laplacian filter itself in the spectral domain by just using as filter rates the eigenvalues of the Laplacian. Or you can make a combination and maybe combine the Laplacian filter with the low-pass filter to have a smoothed Laplacian filter. And infinite possibilities that can be used. Now, to conclude on this, let me also mention a few other very neat reasons to use the graph Laplacian. Probably the most important one is that if you like to measure smoothness of a graph signal, you can actually do that by using the quadratic form of the graph Laplacian. And as you see here, this is the so-called quadratic form of a graph signal with the graph Laplacian, and that is equivalent to taking differences between neighbors on the graph, weighting it with the edge weight, and that is what effectively is computed by the quadratic form. And you can see that this can also be obtained by in the spectral domain by simply weighting the energy of the spectral coefficients with the eigenvalue of the Laplacian, the corresponding Laplacian eigenvector. And of course, as a consequence of that, the smoothness of our basis vectors will be exactly equivalent, of course, to the eigenvalues. And one more remark is that the graph Laplacian also relates to the convex relaxation of the graph of the graph partitioning problem because it's a convex relaxation for minimizing the cut size. And this is related to the so-called Fiedler vector and all algorithms you might know that are called spectral clustering. But I won't say more about that for now. So just to conclude the GSP section, indeed, this has been a very flourishing and emerging field for the last about 7 years. And there have been already a couple of special issues, both in the IEEE Signal Processing magazine and the proceedings of the IEEE, where people are generalizing transforms to graphs, looking at various instances for filtering, sampling, and recovery, and even more recently of proposing graph deep neural networks or to do distributed and asynchronous processing on networks. So if you're interested in this, I really recommend you to look at these special issues to see what's going on. But for now, we know enough about GSP to tackle our human neuroimaging data and to see a little bit what it can do for us. So this has been work that has been pioneered in the lab by Thomas Bolton, who did his PhD in the lab and recently successfully defended it, and also Maria Giulia Preti, who's a postdoc with the CIBM. And the first thing they did is, of course, defining the brain graph. And that is based on structural connectivity by using an established pipeline, the human connectome project data, to have a high quality connectome. And they actually averaged it across 181 healthy individuals. So we have a very, let's say, stable and high quality group level structural connectome defined over 360 regions of interest because we were using the Glacier's atlas. So that's the brain graph. We also have brain graph signals, and that will be our fMRI activity patterns. And we will be considering in this talk today the fMRI that we can get from the HEP in resting state. So let me show you a little bit how the Laplacian eigenmodes look like for this choice of our structural connectome. So as expected, they indeed build up like oscillatory patterns with increasing frequency lambda. And as said, we will use them as the basis vectors of our graph Fourier transform. So here we have the first fellow, which corresponds to lambda 0. It's kind of a DC component, but because we are considering the normalized Laplacian, it has a slight weighting on it. And then we go to number 2 here, which is the first non-zero, smallest non-zero eigenvalue eigenvector. And you see that this basically characterizes the main geometry of the brain, which is splitting it into left and right hemisphere. And we go to the next one, and we see we are basically making an opposition now, anterior versus posterior. Again, very geometric properties of the brain. And we can go on. And of course, we will get patterns that will be increasingly oscillatory as the frequency also increases. And those will be the basis vectors onto which we decompose our fMRI data. So the first thing we did is simply looking at if you apply the graph Fourier transform to the fMRI resting state data, what is actually the graph energy spectral density? So very easily we take each fMRI activity pattern, we compute its graph Fourier transform, we square the spectral coefficients, and we average over all time points. Now, if you plot this in a log-log, this is what you get as a function of the eigenvalue lambda. It's pretty much dominated, the energy is pretty much dominated in the low frequency components, which is very clear because this is a logarithmic axis. So in other words, we kind of see a power law trend that demonstrates that fMRI brain activity patterns that are reflecting function, they actually respect the natural smoothness as this is characterized by the underlying brain connectivity. And that's already a first observation, which is not completely trivial. It's of course very satisfying to see that, but it's not completely trivial because the connectivity between the different brain regions is of course not trivial in terms of, for instance, Euclidean distance, because it's now using all the wiring through the white matter. Still, fMRI is relatively smooth, as you can see, with respect to that structural connectivity. The next operation that I now will introduce, which has been developed by Elvira Pirondini, was also a postdoc in the lab and just recently started a faculty position at the University of Pittsburgh, is to use our graph signal processing machinery to generate surrogate data. And that's of course very useful if you want to do kind of statistical testing and see whether certain types of measures can be explained by null data or not. And now we took inspiration from the famous Fourier phase randomization method that you probably know, typically applied to time series. So you would start off with a certain signal, you do a discrete Fourier transform, and then you apply either phase permutation or randomization into the Fourier domain, and you get back, and you can repeat this many, many times. Each of the surrogate signals will be random, but it's randomized in such a way that it has by construction the same energy spectral density. And by the Wiener-Khinchin theorem, this actually also means that we basically prescribed the covariance structure of that signal. Now we do something very similar, but this time using the graph Fourier transform, where since we don't get actually complex values of our spectral coefficients, but real ones, we only will permute or randomize the signs, and that will be used as an equivalent of the phase in the classical algorithm. So you can again, you take your graph signal, you go into the graph Fourier domain, you permute or randomize the signs, and you get back. You can do this many times with different randomizations. And of course, again, by construction, you will get in the surrogate data exactly the same energy spectral density. And in the original domain, you can easily show, it's a one-liner, that the smoothness as measured by the quadratic form of the Laplacian is the same between the original empirical signal and the surrogate ones. So this can be then applied to our fMRI data. So we, for each graph signal, so for each time point, we consider a graph signal X, we go into the spectral domain, we randomize, and we get back. And you get a surrogate data set, right? And here I have denoted this as a full matrix operation on the data matrix X, which contains all the graph signals in its columns. And P is the sign randomization diagonal matrix. Now, as said, you can do this many times, so you can have you can build a null distribution based on many surrogate sets that can be generated. But actually, one important remark here is that we will always keep for one surrogate data set, we will always keep P fixed. So it means we have the same randomization matrix across time. And that will actually exactly maintain temporal correlation properties. It's again a one-liner to see this. If you actually compute the transpose of your data matrix times itself, which is a T by T kind of a generalized covariance matrix, if you like, you see that this is identical to this T by T matrix you would get for the empirical data. So in terms of temporal structure, this is fully maintained in the surrogate data. So in other words, this leads to pretty strong null data because it has the same smoothness in space and it has exactly the same temporal correlation properties in time. Now, a final ingredient I need before I can show you some nice application is that we also like to have a null model for the structural connectome, just to be able to kind of knock out the influence of the specific structural connectivity that we measure in the brain. And for that, we're going to choose the configuration model, which is a very simple null model that conserves the degree distribution, but without any preferential connectivity at all. And mathematically, it is constructed as the outer product of the degree vector with the proper normalization, basically spreading out the total edge strength of each node across all possible, across all other nodes. So to visualize this, here you have the original structural connectome, and this is the configuration model computed out of it. Okay. So what we do now next is defining our GSP filtering operations. And for that, we will make a choice of, we will define the filters by their spectral windows. And we will go quite simple. We actually choose an ideal low pass and an ideal high pass filter. And to do that, we look again at our averaged graph spectral density, energy density that we got from our fMRI data. And we're going to decide on the cutoff between for the low pass and the high pass filter as the frequency for which we have equal energy on both sides. So it's basically making it such that we have on average as much energy here in the low pass area, which only needs about 30 graph frequency components, and we have then the high pass filter, which takes the rest, which is 330 frequency components. So you see that to have equal energy, we only need 10% of the total amount of modes. And so the low pass filter will just be an ideal low pass filter like this, and the high pass filter will of course be like this. Now, why do we do that? Because we will interpret our low pass filter as one that will take out signal that is aligned with the underlying brain structure or coupled with the brain structure. And the high pass filter will be emphasizing activity, so patterns in the graph signals that is more liberal or decoupled with respect to the brain signal. So that is the interpretation that we give to respectively the low and the high pass filters. So then you can apply this. We have here our original fMRI data. For each time point, we have our graph signal. We go into the spectral domain, and we have our ideal low and high pass filter. We have the aligned and the liberal part, and we can get back to the original domain to get the filtered versions of our fMRI dataset. And next, and this has been a great idea by Maria Giulia, we define the structural decoupling index, which is the ratio of the liberal versus the aligned energy in a specific node. So we have these two filtered data sets of our original fMRI, and now we're going in each and every node, we're going to compute the ratio of the energy that we get in the liberal versus the aligned version of our filtered version of our fMRI. And that will define the structural decoupling index. Now, since we have chosen the cutoff such that on average the energy is equal in the low and in the high pass part, you might expect that probably this decoupling index should vary around 1. And I'm going to show you in a moment, but then we're going to do that not only on the empirical data, but also on surrogate data, either surrogate data generated on the empirical structural connectome or surrogate data generated on the configuration model. Okay, so if you apply the structural decoupling index to the surrogate data that is generated on the configuration model, but analyzed with the structural decoupling index that is using the information from the empirical structural connectome, this is what you get, a very red pattern, meaning that all nodes are very much decoupled or liberal with respect to underlying structure. And that makes, of course, perfect sense because the surrogate data didn't respect the smoothness that is available or that is in line with the empirical structural connectome. Now, if we generate the surrogate data also using the empirical structural connectome, we get a completely different pattern, as you can see here. We get a very characteristic tendencies which show us that sensory regions are very much towards coupling. So they are very much aligned with underlying structure. While if you go to more high-level regions, frontal, temporal pole, and so on, they tend to be more decoupled, so to be more liberal with respect to underlying structure. And that's a very clear pattern that we get already using our surrogate data. Now, if we compare this with the structural decoupling index applied to the functional empirical data, this pattern actually reinforces itself. So you see that now the decoupling in the sensory regions is even stronger. You can clearly identify the visual, the somatomotor, and the auditory regions. And you also see that the decoupling in the high-level regions, now also highlighting parietal and again frontal and prefrontal regions, is getting stronger as well. So what this shows us is that structural decoupling is confirming this well-known hierarchical organization from the sensory or unimodal regions to the high-level cognitive ones, which are supposed to be, of course, much more transmodal. And this is a result that has been reported using brain gradients from functional connectivity, but also using other types of measures such as cortical microstructure, gene expression, and neural temporal properties, always showing this very same hierarchy from unimodal to transmodal regions. Now, what is a bit striking also is that this hierarchy is already present when we use the surrogate data. It's of course stronger when we use the empirical data, but already for the surrogate data, we can clearly identify this hierarchy, which probably in a way means that if we have appropriate smoothness of activity patterns, that is actually sufficient to let this hierarchy emerge as characterized by structural decoupling. So let's now look a little bit closer at the difference between the structural decoupling index on surrogate data versus empirical data. And here is the computed difference. And so what you see here is that indeed, in most sensory regions, the empirical data is actually more coupled, except for the visual region. So there seems to be a certain opposition between visual and the other regions. Otherwise, all the high level, if I call them like that, or transmodal regions, they are for sure more decoupled in the empirical data than in the surrogate one, which is of course a good sign. Because this means that from one hand, we see that the structural decoupling difference is actually in line with this secondary gradient that is reported from these previously mentioned functional connectivity gradient studies and also in the genetic studies. So the opposition that we see between the visual and the other sensory regions is something that we observe in our difference between empirical and surrogate data. And also the more strongly decoupled regions, frontal, parietal, so the transmodal ones, or the ones that are commonly reported as being much more specific to an individual, as for instance in applications such as fMRI fingerprinting. And something very nice is also when we replicate the link with behavior using the same analysis as proposed by Daniel Margulies in 2016, by linking it with neuroscience, we indeed see that we also reveal using the structural decoupling index a gradient in the brain that clearly goes from more low-level processes to high-level ones, very similar to the one that was revealed by Daniel in his paper using functional connectivity gradients. So in other words, it seems to be also a behaviorally relevant gradient as well. Okay, let me quickly the time. Okay. So the next part of results will be on high-resolution brain graphs. So this is work that is spearheaded by Hamid Bejat and Anjali Tarun. Hamid is a collaborator at Lund and currently a postdoc at Harvard. And Anjali Tarun recently finished her PhD in the lab and will actually, in 2 weeks, have her public PhD defense. So the idea here was to build up a very high-resolution brain graph. This time not using S-nodes brain regions that have typically several hundred to several thousands of voxels, but considering as a node of the graph each and every voxel, not only in the gray matter, but also in the white matter. And if you do that at the typical resolution of which we have the diffusion-weighted MRI, like from the HEP, that leads to almost 1 million nodes in total. So this whole operation is done in subject space, and we are using extracting from the diffusion-weighted MRI both information about the magnitude and about the orientation, and combining that into local connectivity between the nodes of the graph. So this leads to a huge but very sparse adjacency matrix, because you basically only have potential connections between neighbors. What is really key is that this graph, although it looks maybe at first hand cumbersome, and it is in a way, but it is encoding actually physical distance in the number of hops that you would do if you travel on the graph. And that is something that is lost if you go to the simplification of the structural connectomes as I've used them in the previous part. And we will see why that is important in a moment. Even if those graphs are huge and should be stored as sparse matrices, you can actually still compute quite some leading eigenvectors using common large-scale numerical solvers. And actually, if you do that, and we have several designs for the brain graph depending on how you extract magnitude and orientation information, but if you look at the leading Laplacian eigenmodes, so the ones with the smallest eigenvalues, you get these very nice brain maps, because this time you can directly visualize, of course, your eigenmode into a volume, because there is a one-to-one correspondence between a voxel and a node. And you see that we get a very nice general, let's say, DC component, which now runs through the white matter. And then the first smallest non-zero eigenvector shows again a more geometrical pattern, this time showing first the anterior versus posterior contrast before going to the left versus right hemispherical contrast. And I can show you a few more. Here you start to have combinations highlighting the cerebellum in particular versus the rest of the brain. And then you have like crossovers between left, right, and anterior, posterior, etc., etc. But what you can see is that we have always very nice structure that is highlighted within the white matter next to covering the gray matter as well. And what you can then do is, for instance, look a bit closer at these Laplacian eigenmodes and look how much they overlap with white matter bundles that you can get from a white matter atlas. And so you can, for instance, have a kind of a matrix like shown here, which tells you whether a certain tract is positively or negatively contributing to specific Laplacian eigenmodes. And although I don't have time to go much into detail, but as said, all these high-resolution brain graphs are made for are subject-specific, and we actually also looked how to normalize them and come up with a group-level representative, and that is showing that these very high-resolution brain graphs show a consistency between subjects for a dimensionality of approximately 300, which in a certain way is bringing you back to the typical kind of numbers you would get in brain atlases, but now completely driven by the connectivity as you get it at the high resolution. And so then Anjali used this in her PhD to look into what I call inpainting of brain activity. What is it about? So if you now on this high resolution brain graph represent the functional bolt signals on the gray matter, you might get something like that if I'm looking at a single axial slice. And when we are going to combine that with our brain graph, what we're going to solve now is a problem of coming up with biologically plausible activation patterns on the white matter. So we're not going to try to recover activity from the white matter. We're just given the activity on the gray matter and given the connectivity on the white matter, can we come up with what could be a plausible activity pattern that matches both? And the ultimate idea is that this might help us to get insights in how brain activity is mediated by the underlying structure. So this again can be solved by graph signal processing, and it's a graph signal recovery problem. In a certain way, the inspiration also comes from the boundary value problem of heat diffusion. So you can consider the gray matter as a plate where you have a certain pattern of heat pattern, and that is now diffusing inside the brain on the white matter structure. So you have this heat conduction throughout the structured white matter. And if you write this down mathematically, this is about recovering a smooth signal on the brain graph, gray and white matter, where you have a certain data fitness, which is only enforced on the gray matter, but we also want to, the finally recovered signal, we want it to be smooth on the whole brain graph. And solving this can be most easily done using an iterative implementation given the size of the graph, where we basically do an update step together with the diffusion step to ultimately get to our inpainted brain volume. And of course, the regularization parameter needs to balance between the data fit and the smoothness and can be optimized by established methods. So here you see a little movie of how in-painted fMRI data looks like. I hope it comes through WebEx, probably a bit shaky. But it is showing you the gray matter extrapolated signal on the white matter as you go along. And it's maybe important to say that the way we solve the in-painting problem is different from tractography because it's the whole pattern on all of the gray matter that makes up the boundary conditions and that will actually influence what is recovered on the white matter. And so there is a dependency, if you like, on the complete pattern on the gray matter and not just, you cannot just break this down and for instance, start coloring tracts. This would not be equivalent to what we solve. And of course, the recovered graph signals can be directly embedded in the volume, as I already highlighted before. So then Anjali wanted to use this to look into brain dynamics. And the first thing she did is looking at PCC seed connectivity. So here you have on the original fMRI data, using the PCC as a seed region, the connectivity map over resting state fMRI. And so you get the typical default mode task negative versus task positive contrast, as we all know. But if you apply this very same PCC seed connectivity to the interpolated or in-painted fMRI signals, you get something that is very similar on the gray matter, of course, but that also shows you which white matter tracts, white matter bundles, are heavily loaded to explain the given observations of activity on the gray matter. And you see that various known, in this case, well-known white matter tracts are indeed engaged to explain our PCC seed connectivity pattern. And it's very nice to get this kind of result without an explicit, for instance, thresholding of the original map followed by tractography. This is kind of fully data-driven by the original data and the in-painting of it. You can also look further into more dynamics by, instead of looking at the PCC seed connectivity map, you could do a PCC caps approach, which if you apply this PCC caps approach to in-painted data, you get to what I call in-caps, and which show you for each cap that is occurring how the white matter is mediating the distributed gray matter activity that we observe. And here is just a little zoom up of one cap where you can see that indeed, again, many tracts are supporting the gray matter pattern that we see. And if you like to see this in more detail, you could now, just as we did with the Laplacian eigenmodes in the very beginning, we could compare the in-painted caps in terms of their white matter loading with respect to a white matter atlas. And you see that the first row here is the loadings of the tracts on the seed connectivity map. And you see that indeed the dynamically occurring in-caps are using the white matter in a much more distributed way. And that's, of course, very interesting to now look more closely and to see also how the mediating white matter is actually supporting those dynamically occurring coactivation patterns. Okay, and then I need to speed up a little bit. In a very recent application, she is actually using, Anjali is using the diffusion time as a proxy for distance. So we start off with the seed connectivity map established on the gray matter. We get our in-painted map out of that. And then we're going to, alternatively, to simply diffuse the seed connectivity map on the gray matter until we establish basically what is the best match with respect to the inpainted map. And why we're doing that? Because in this case, we're using diffusion as a proxy for distance. So diffusion time becomes a proxy for distance because, as said, physical distance is embedded by hopping on this voxel-wise brain graph. And very nicely, if you now make this comparison, you see that there is a very clear phase transition in terms of similarity between the inpainted map that has the full information and the best match that happens at a certain diffusion time. And so that can be translated in what we call a structure function range map. And in this case, we started from regions defined by the EU17 atlas. And we translated already the optimal diffusion time in the corresponding anatomical distance. And you see that you get a very nice map again, highlighting actually again a range of functional interactions that is respecting the cortical hierarchy, as I already showed you before, going from unimodal short range to transmodal long range. And it's also nice to compare this against the geodesic cortical distance maps reported by Daniel Margulies, also in his 2016 paper. And just as a note here, if you would do this with simple Euclidean distance in the volumetric way, you would get a much more messy view on structure function interactions, which is here reflected by the scatter plot between the anatomical range that we get from our method and simple Euclidean distances that would be computed in the volume between the gray matter areas of a connectivity map. So let me conclude a couple of minutes with ongoing work by Milan Petrovic and Raphael Liégeois, and also Thomas Bolton, who contributed more recently. And so this is about community-aware graph signal processing. So what is this about? So I already kind of told you that we are all very happy to use the graph Laplacian, and that it's great that this is providing an equivalent of the Fourier transform to be applied to graphs. But actually this whole intuition breaks down if the graph becomes what I call too irregular. And to give you a couple of examples, if you have a completely connected graph, let's say one that is overconnected, then all eigenvalues are n except one that is 0. So this is obviously not a very good graph Fourier transform. If you have a star graph, which you can consider as a graph which is interconnected, then one eigenvalue will be 0, one will be n, and all others will be 1. Again, not very great as a graph Fourier transform. And there are many other cases that have been described in mathematics. For instance, if you have star-like trees, you have a certain phase transition phenomena that happens, where you can also prove that many eigenvectors will be actually localized eigenvectors, which again is not something that is making up a great graph Fourier transform. And so another type of graph that is typically not so well suited to be analyzed with the graph Laplacian are graphs that have very strong community structure, meaning that they have dense subgraphs that are well separated between them, such as, for instance, this collaboration network that you've probably seen if you are interested already in this topic. Now, why am I saying that? Because, as you might know, if you want to look for a community structure in a graph, then the modularity index is one measure that is commonly used. And actually the modularity index is quantifying the density of subgraphs, comparing it against the null model, which is the configuration model, the very same that I used before. And the great thing here is that the modularity index can also be seen as a quadratic form of a modularity matrix. So in other words, you can think about community graphs and operators that characterize community structure by thinking about a modularity matrix, which is the adjacency matrix minus the configuration model. And as a matter of fact, the eigendecomposition of Q is also solving the convex relaxation problem in community detection. Okay, so as a final comment here is that what we're currently doing in the lab is defining community-aware graph signal processing filters. Because instead of using the Laplacian spectrum, we can now go with filters defined on the modularity spectrum. That could be, for instance, modularity promoting, if they are in this part, or they could be modularity neutral, or they could be anti-modularity promoting filters. And Thomas has been working on one application where he's trying to link these filtered signals in the various forms, either using the Laplacian low and high pass or the modularity and the anti-modularity, to different behavioral scores from the HMP database. And so to make a long story short, what you can see by increasing the bandwidth from one hand and by picking different types of filters, either Laplacian or modularity based, you can see that you get strong associations. These have been corrected by Bonferroni corrections, and that actually many associations, most associations are recovered using the modularity based filtering. And so we are currently looking into why exactly this is the case and what properties in terms of GSP are we actually picking up with these filters that are a bit different from the Laplacian structure. So let me conclude. I hope I convinced you that GSP is a versatile new framework that can be applied for computational neuroimaging. It can be used in many settings, different scales. And it's a principled way to define new operations. If you go with the Laplacian, you're defining your operations by smoothness and gradients, or with modularity by community structure. And in its application, we've seen that it can reveal unimodal to transmodal hierarchy in the brain, either using, for instance, our function-structure relationship or the functional interactions range. And of course, now the big question in the future will be, can this also be used to understand perturbation during task or in disease and disorder? And one of the open challenges here is also to link GSP more closely with temporal dynamics, because what I showed you today was actually only using spatial type of GSP, and this can be further generalized and linked for instance, with biophysical and neurocomputational models. And to conclude, let me thank again the whole lab, in particular the people who contributed particularly to the GSP work in the group that I already mentioned, and also the collaborators. And thank you very much to everybody. And sorry for going a little bit over time.
Thanks, Dimitri. That was great. So do folks have questions? Let me see here. Please go ahead and ask your question in the chat if you'd like. I see that we have one here. Just a minute, I'm just trying to pull it up. So one of the questions is, I think you are implicitly assuming edge weights are non-negative. Why not negative edges to denote anti-correlation or dissimilarity?
Okay, so in everything I used here for GSP, the graph itself was always a structural graph. So there were no negative edge weights because this was edge strength as being structural connectivity. Now, more generally, of course, you could think about a GSP application where you do use a functional connectome as well. And in that case, you need to think a little bit more closer. There are already extensions. I mentioned them on the slide of the GSP work in the IEEE realm. And there are already extensions proposed to define properly Laplacians for signed graphs. Okay, but this is an ongoing work where several proposals have been already put forward. In our application here, we didn't have negative weights.
Okay, thanks. And that was a question by Jean Shung, by the way. Now, Thomas, the Atlanta Thomas, says that there's work finding surface parcellations, e.g., or i.e. desiccant and HCP, that don't perform well compared to volume-based parcellations for tractography, because tract accuracy tanks when the streamline hits the gray matter, white matter barrier. has your group explored that in your models?
No, so not yet.
Okay.
Yeah.
Okay.
Yeah.
Feel free to ask any other questions. We're right at 12, but we can do maybe one or two more while we're waiting.
So
yeah.
there's a few questions in the chat panel, if you may ask.
Oh, yeah. Great. Okay.
So I don't see the chat, so I'm sorry if you...
That's fine. I'll read it to you. Don't worry about it. Let's see. So yeah, I think this is really interesting work, and I'm looking forward to seeing how you extend this to the temporal dynamics in the functional data in particular. And then related to that, one of the things that we've been looking at more recently is spatial variation, right? Spatial variation in nodes, across subjects and over time. And so that's yet another layer of complexity, but these approaches could certainly be extended into those areas, I think.
Yeah. And some of the work that I mentioned in the beginning of other works that are looking at links between structural and functional connectivity, like the structural connectome harmonics and the brain activity eigenmodes, they're actually mostly inspired by starting from the dynamic viewpoint. So they want to see this as the best basis decomposition for dynamically occurring activity. And for instance, the brain activity eigenmodes, they are defined by decomposing the activity in a dynamic way, right?
Right.
So there have been already extensions, but they have not been linked within this framework of GSP. So there are similarities and there are differences.
I think the one twist on that that might be interesting would be to again look at spatial modes of decomposition that then vary, right, over time as well. So there's not been a lot of work, I think, in that area, but there has been some, of course. So
yeah. In our case, the decomposition is completely time independent, right? So it's and it's even the same structural connectome in the examples I've shown you for each and every subject. So this is another extension we're working on now, is to see if we have both subjects, structure that varies and function that varies, what does this bring in addition, right?
Okay. So thanks again for joining us. So for the Brain Space Initiative, what we will do is we'll create a Slack channel for further discussion. So if people have questions and the like, we can kind of engage there. And we'll also have the video released there as well. And Armin, did you have any other comments?
So there are a few questions in the chat panel that we can answer if you want in the Slack, or we can just go over them quickly. So
it's fine for me.
So Yuping is asking, what is your thought for combining multi-graph signal with GFT?
I think those chats went right to you personally, Armin. That's why we can't see them.
No, okay. I think we should be able to see, but so the question is, what is your thought for combining multi-graph signals with GFT?
Which type of signals? Multi-graph signals?
Multiple graph signals, I guess.
Multiple graph?
Yeah.
I
don't know in which sense this question is meant. I mean, now we are decomposing, we are applying, for instance, the graph Fourier transform to each and every time point, right? So there are multiple ones. I don't know what is actually meant with multiple specifically here, because we already.
So maybe I can unmute Yuping if he wants to ask his question himself. Yeah, Yuping, would you like to ask your question yourself? Is unmute right now. You're on mute.
Oh, okay. Can you hear me?
Yes.
So actually, I'm attending another talk. So so the multi-graphs, for example, you can derive the graph from different tasks, or you can get the graphs at different resolutions, because of the use of, different passation schemes. So how to combine those, multi-graph signals from different resolutions, from different tasks, or
okay, sorry. Yeah, I get it. So with respect to porcellations, we already either adapted the glacier, for instance, by adding regions which were not well represented, or taking other porcellation schemes. And actually, the gradient that I showed you from the sensory to the high-level brain regions, we always find it over and over again. So that seems to be very robust. For characterizing structural decoupling index. So that's good news. With respect to different tasks, because all this was resting state. So this is currently ongoing work by Maria Giulia, where she's computing the structural decoupling index, not only for resting state fMRI, but also for different tasks like the ones we can find in the HCP. And the general pattern, so if you would just look at the general map, it's always the same. It's always this very same gradient from sensory to high-level regions. So visually, you wouldn't see anything. But if you analyze this more closely, you see that we have very consistent small differences between the different tasks. So indeed, the SDI is picking on top of this general gradient differences that are characteristic for the different tasks. But this is ongoing work by Maria Giulia, so it's not yet published.
Yeah, get it. Yeah, so I guess the graph signal processing can offer a appealing approach to identify common network structures among those multiple graphs. At the same time, they can also identify graph-specific structures, which may reflect some structures in the brain connectivity network. Thanks.
Great.
Okay.
So, Armin, do you want to maybe ask one or two more questions? I think you can see them.
Sure. So the next question is from Ray Duran. He's asking if there is any research into correlating fMRI work with calcium imaging in your group, probably.
Yes. So I mean, it's not related to GSP, but in other work in the lab, we have been looking at dynamics and what we called total activation, which is a deconvolution, a regularized deconvolution approach. And that is actually driving what we have also called innovation-driven coactivation patterns, or ICAPs. And so that work has been recently extended by Younes, who's actually on the picture over here, by Younes for calcium imaging in zebrafish data. So also brain activity, but this time using calcium reporters. And he used actually extended the same framework, but now instead of plugging in there the hemodynamic response function that characterizes the system, he uses a calcium response function. And then applies this to the zebrafish functional data, and indeed shows that you can also establish very reliably maps of transient activity and cluster those maps into what you could call ICAPs for zebrafish. So this has been done quite recently, and it's a paper in Transactions on Medical Imaging, if you're interested.
Let's hope the ICAPs are waterproof.
Indeed. They even work for fish.
Yeah.
So Kamal has two questions. First is, with GFT, are we assuming that graph topology is invariant? If yes, how useful would it be for analyzing real world network where edge can come in or vanish? And he's also asking if these data sets are publicly available or not.
Yes, so very good question. So for now, we assumed that indeed the structure is fixed, right? We even assumed it to be fixed between subjects. If you will make it, for instance, subject specific, it also means that you will, the whole definition itself of the graph Fourier transform will actually slightly vary between your subjects. And so then you need to choose, I mean, either you find an interpretation on why Laplacian eigenmodes are not exactly the same between your subjects, or you come up with a kind of a jointly determining maybe the best matching GFT between all your different subjects, which is, or all your different graphs in that case, which is something that is also available in the literature with algorithms that have been developed and could be done. So like, let's say the best common denominator in terms of a graph Fourier transform between your different subjects. And there are many other choices you could make. So now about, very good, you brought up the availability. So all the source code is available on Git repositories. For instance, if you look up the link in the paper on the structural decoupling index, you can download the scripts, and the data itself is HEP, so that's also publicly available.
Yeah, that was also a question from Yunus. He was asking, there are like not a limited number of available library, and he was asking about the coding platform and also the code if you have available.
Yes, it is available.
So, and the other question is from, I think, Irat, regarding the eigen decomposition in the first part of the lecture. The first one highlighting the whole white matter structures are quite intuitive. Could you elaborate on the possible meaning of cross-highlighted?
Of cross-highlight?
Highlighted. So it's like that.
Okay, wait, let me maybe. So I guess it's about.
And I'm also going to unmute Irat if he wants to ask him a question himself or follow up.
So I guess it's probably about these patterns that occur here. So
I think
yes, yes, sir, you're right. So
okay.
The second row, the middle picture.
So I don't have a specific interpretation for these because it's just like the Laplacian eigenmodes that we had on the course structural connectome. Is these first ones, they clearly capture general geometry of the graph. And it seems that general geometry on the brain graph is actually in a certain way quite similar to what we get on the coarse structural connectome, being left-right hemisphere, anterior-posterior, and then combinations of those always getting to finer and finer regions, if you like, because somehow let's say the oscillatory pattern is increasing in frequency. So I don't have an explanation why exactly, for instance, Laplacian eigenmode number 5 would need to have this shape. But it is building up the basis on the principle of Laplacian smoothness of the Laplacian. So that is actually the most smoothed solution you could have if you have already established the 4 previous Laplacian eigenmodes. So this is the most smoothed next one that is orthogonal to all the previous ones.
Okay, thanks, sir.
So there are, well, I have one question myself. So I was wondering if you have looked at the structure reconstructing this, applying this Laplacian eigenmode and others on the whole brain voxel-wise functional connectivity in the subject level. I've done a study on the group level. I can extract the eigenmode, very nice eigenmode from at the voxel level from functional connectivity. But I was wondering how, or what your thought is on like either window or like subject level.
So we didn't do anything like that, right? For us, activity was always considered as the activity patterns. So they state, let's say, they were our graph signals, and they were never reduced to graphs that reflect functional interactions. Because that's the way we thought for a first application of GSP was the most logical thing to do. So the graph is the structure and the function is the graph signal. But now, yeah, you could go different ways. And what you're suggesting would be, I guess, very close to methods such as the diffusion embedding as it has been used by Daniel Margulies and also connectopies by Christian Beckman and many others, where you basically could establish gradients or clustering segmentation based on long and short range functional connectivity, because I guess that's what in the end you could do with those. So yeah, there has been work already in that direction, but we didn't use functional connectivity in a setting of graph signal processing. I mean, to define the graph, right? Only to.
So, and maybe the last question from Adan Hanif, he's asking, she is asking, I'm not sure, what are the future interesting prospects that we can see or work in the domain of brain graph neuroimaging and GSP in general?
Yes, so I think if I go back, I was probably going a bit too fast at the end. I mean, it's related to what I've put here under challenges, right? So there is, for sure, linking GSP more closely with temporal dynamics as a general theme, because we basically just computed then energies after doing GSP filtering, we just computed energies over time. So you can do, of course, much more interesting stuff. Now, there has been already in the GSP field graph models that could be very interesting to incorporate within a spatial graph, as I've shown you, also kind of by a tensor product, a temporal graph. Okay, so conceptually, this would be a huge graph because each node would be represented for each separate time point, but it's not necessarily needed to define that graph explicitly. So you could come up with graph signal processing operations that implicitly are using that very huge tensor graph, but that can still be implemented at the end as in a very efficient way. So there have been papers on that in the GSP field that perhaps could find their place also to be applied to neuroimaging. And then I think a third point is making connections with the work that has been done on the biophysical and the neurocomputational models that I briefly mentioned in the beginning, because obviously that would also be very nice to have maybe a graph Fourier transform that can be more closely linked to neural dynamics as you could get from those models as well, right? And yes, I mean there are many directions one could go there.
Mm-hmm, mm-hmm. Great.
Thank you. I think I cover all the questions in that channel.
Okay. Well, I wish we could take you out to coffee or something, but we'll plan for doing that later. Thanks so much for joining us, Dimitri, and let's stay in touch virtually and continue the discussion in the Slack group and elsewhere. So thanks so much.
Thanks a lot, Vince, and it was really fun. So thank you also everybody for attending.
Hopefully it wasn't too painful talking into a void like that. So
you're getting used to it.
Yeah.
So maybe I just one question for the Slack group. Could you send me an invitation link for that?
Sure, I will send you. I sent before, but I will definitely make sure. I will send it again just to make sure.
Okay. Sorry, I think I.
And if there is any problem, please let me know via email.
Okay.
I will fix it. Thank you.
Very good.
And thanks everyone for joining. We really appreciate your participation. So.
Thank you.
All right.
Have a nice weekend.
Everybody. Okay.
Bye-bye. Take care. Bye-bye.
Virtual wave. Okay. Bye.